So this week we will continue on chapter 3, photosynthesis, on the last subtopic, which is C3, C4, and CAM plants. So what are C3, C4, and CAM plants? So C3 plants are the one that we learned previously, which is about the light reaction and also dark reaction, or also known as Kelvin cycles. So in C3 plants, the initial fixations of carbon from carbon dioxide is via Rubisco, in which the Kelvin cycle, the enzyme that aids carbon dioxide to ribulose biphosphate. They are being called as C3 plants because the first organic product of carbon fixation is 3-carbon compound, which is 3-phosphoglycerate, or also known as glyceraldehyde 3-phosphate, from 3 molecules of carbon dioxide. So this plant produces less food when the stomata close in hot and also dry days, such as the rice, wheat, and also soybean. For C4 plants, they are being called as C4 plants because they preface Kelvin cycle in an alternate mode of carbon fixations that form a 4-carbon compound as its first product of carbon fixations. So in C4 plants, there are 2 distinct types of photosynthetic cells, which is bundle sheet cells that being arranged into a very tightly packed around the vein, and also mesophyll cell, which is the structure is more loosely arranged. So C4 plants minimize the cost of photorespiration by incorporating the carbon dioxide into 4-carbon compound in the mesophyll cell. So this step requires the enzyme of PEP carboxylase. So the PEP carboxylase has a higher affinity for carbon dioxide compared with Rubisco. Therefore, they can fix carbon dioxide even carbon dioxide concentrations are very low. So these 4-carbon compounds are exported to bundle sheet cells, where they will release the carbon dioxide that is then used in the Kelvin cycle. So example of C4 plants are sugarcane and also corn. So as you can see here, in C3 plants, the bundle sheet is over here, and this one is the mesophyll cell. However, as you can see in C4 plants, the mesophyll cell surround the bundle sheet cells. So therefore, C4 plants have a crunch anatomy in which the mesophyll cells are usually arranged in a circle around a bundle of sheet cells to ensure that the product of the carbon fixations in C4 plants, which is the mallet, can easily pass to bundle sheet cells to undergo the process of Kelvin cycle. So this is another clear figure of how does the C3 leaf and C4 plants look like. It's quite different. As you can see, the mesophyll in C4 plants are arranged in a circle and surrounds the bundle sheet cell that surround the vein. So for C4 plants, the first carbon fixations occur in the mesophyll cell, in which it will cooperated with PEP carboxylase instead of Rubisco, and then the 4-carbon product, which is the mallet later, will go to the bundle sheet cell to undergo Kelvin cycle to produce the sugar. So let's look more detail on the C4 plant pathway. So PEP carboxylase, as you can see here, has much higher affinity for carbon dioxide compared to Rubisco, and PEP carboxylase has no affinity at all for oxygen. Therefore, no photorespiration will occur. So PEP carboxylase will add carbon dioxide to phosphoenol pyruvate PEP, and it will form oxaloacetate, which is a 4-carbon compound. So oxaloacetate are the first product of carbon fixation. That's why, and it has 4 carbon. That's why this plant's name is a C4 plant. And then oxaloacetate will reduce to form a malate, in which they will use an ADPH that being produced during the ligation, and then the malate later will send to the bundle sheet cell through the plasmodesmata. So the malate later will be oxidized and decarboxylate to form a pyruvate over here, and also to form 1 molecules of carbon dioxide. So the pyruvate, okay, will enter back the mesophyll cell, and it will be reduced, and it will receive phosphate group from the phosphorylations of ATP, thus producing back the phosphoryl pyruvate. Meanwhile, for the carbon dioxide, it will go to the Calvin cycle, as it will bind to ribulose biphosphate and being catalyzed by the enzyme of Rubisco. Okay, so and this Calvin cycle happen in the bundle sheet cell, thus producing a sugar that will export into the vascular tissue, either in the phloem to transfer to the other parts of the plant. And you have to remember that bundle sheet cell contain only photosystem 1 to carry out the cyclic electron flow. So what are the advantages of C4 pathway functions in addition to C3 pathway? So the first one is that photosynthesis can progress even at a very low carbon dioxide concentration in C4 plants. This is because of the great affinity of the PEP carboxylase toward the carbon dioxide instead of towards the oxygen. Therefore, this will allow the photosynthesis to occur even when the concentrations of carbon dioxide is very low. The second advantage of C4 pathway is that it can store carbon dioxide temporarily, so it can be used later. So the carbon dioxide that is released from the decarboxylation used in the Calvin cycle, and the extra carbon dioxide that will accumulate inside the cell and being stored by the C4 plants, and it will be released when the supplies of carbon dioxide from the environment is being reduced. And last but not least is that C4 pathway can avoid the photorespiration. This is because they have PEP carboxylase that more having a great affinity toward the carbon dioxide, thus prevent the photorespiration to occur and making photosynthesis more efficient. As you know, photorespiration process is a wasteful reaction. Last but not least is that CAM plant. They are being named as CAM plant because this type of plant use crassulacean acid metabolism to fix the carbon. So what happened? CAM plant is exactly the same with C4 plant. However, the time of the carbon fixations to occur is different. So in CAM plant, the stomata will open during the night, therefore incorporating carbon dioxide into organic acid. And during the day, the stomata will close, okay, to avoid from the losing of the water from the plant. However, the carbon dioxide will be released from the organic acids during the night just now, and during the day it will be released from the organic acid and being used in the Calvin cycle. So the CAM plant is similar to C4 plant where except except uh, except for not except yeah except for the side of the Calvin cycle, because C4 plants occur in bundle sheet cell. Meanwhile, CAM plant, the carbon fixation only occur in mesophyll cell. There is no bundle sheet cells um, involved in CAM plants. So example of CAM plants such as cactus, aloe vera, and also pineapple. So in CIM plant, during the night, the stomata will open and PEP will incorporating with carbon dioxide to produce oxaloacetate, in which later will be reduced to form the malate. The malate will be stored in the cell vacuole at night and being converted into the organic acid to prevent the pH changes in the cytoplasm. And then during the day, the stomata will close. The malate that in the cell vacuole will be oxidized and decarboxylated to produce the pyruvate and also carbon dioxide. So the carbon dioxide is released from the malate that will later be used in the Calvin cycle. So the pyruvate is phosphorylated to regenerate the phosphoenol pyruvate to bind again with the carbon dioxide. So the concentration of carbon dioxide is increased in the mesophyll cell, and they can prevent the photorespiration from happening. So as you can see here, during the night, the stomata will open, and then they can uptake the carbon dioxide, and carbon dioxide will incorporate with the PEP producing oxaloacetate, and then oxaloacetate is being reduced to form malate, in which the malate will be stored in the cell vacuole as a malic acid. So during the day, the malic acid inside the cell vacuole will be converted into malate and released from the cell vacuole, and then the malate will form carbon dioxide that will undergo the process of Calvin cycle, and then also it will form the pyruvate, in which the pyruvate will be reduced to form PEP phosphoenol pyruvate to bind again with the carbon dioxide during the first carbon fixation. So as you can see here, the C4 plants and CAM plant, the carbon fixations is the same. They have two carbon fixations. However, carbon fixation of C4 plant occur in mesophyll and bundle sheet cells. Meanwhile, in CAM plant, the carbon fixations only occur in mesophyll cell. So let's look on the differences between C4, C3, and CAM plant. C4 is C3 known as C3 plant because the first product of carbon fixation have 3 carbon, and C4 plant, the first product of the carbon fixation have 4 carbon, which is oxaloacetate, and for CAM it is called as a CAM because they are using cresolution acid metabolism to fix their carbon. So example of C3 plant, tomato, wheat, and rice. For C4, such as the sugarcane and also corn, and CAM, such as the cactus and also pineapple. So in C3 plants, the carbon fixation only occur once, which is during the Calvin cycle. Meanwhile, the C4 plants occur twice, which is the first one occur in mesophyll cell, the second one occur in bundle sheet cell, and then for CAM, the carbon fixation also occur twice, same with the C4 plants. And then the site for carbon fixation and Kelvin cycle for C3 plant, the Kelvin cycle, which is referred to the carbon fixation, occur in mesophyll cell. Meanwhile, in C4 plants, the first carbon fixation occur in mesophyll cell. Meanwhile, the second carbon fixation, which is the Kelvin cycle, occur in bundle sheath cell. And for CAM, both of the carbon fixations and also the Kelvin cycle occur in mesophyll cell. However, the carbon fixation occur at night when the stomata are open, and Kelvin cycle occur during the daytime when the stomata is closed. So the time of carbon fixation for C3 plant is during the daytime. For C4 plant, also during the daytime. Meanwhile, for CAM, the first carbon fixation occur at night when the stomata are open, and the second carbon fixation, which is the Kelvin cycle, occur during the daytime when the stomata is closed. And then continue. The first carbon acceptor in C3 plant are ribulose biphosphate. Meanwhile, in C4 and CAM plant are ribulose biphosphate and also phosphoenol pyruvate. For carbon dioxide fixing enzyme, C3 plant is Rubisco. Meanwhile, C3 plant is Rubisco, which is you can find it in the Kelvin cycle. Meanwhile, C4 and CAM plant are Rubisco, which is you can find it in the Kelvin cycle also. Meanwhile, PEP carboxylase, you can find it in the first stage of carbon fixation. And the rate of C3 plant is very high because Rubisco have higher affinity towards the oxygen. Meanwhile, for C4 and CAM plant, the rate of photorespiration is very low because PEP carboxylase have higher affinity towards the carbon dioxide. The rate of respiration and photosynthesis in C3 plant is very low due to the photorespiration process. And C4 and CAM plant, the rate of photosynthesis is high because the rate of photorespiration is low. Therefore, C3 plant, the yield is low due to the rate of photosynthesis, and C4 and CAM plant, the yields are high due to the higher rate of photosynthesis. So now we're gonna look on the factors that affect the photosynthetic rate. The first factor we're gonna look at is the light intensity and the wavelength, and the others are temperature, concentrations of carbon dioxide, concentrations of chlorophyll, concentrations of oxygen, water, and also chemical compound. So for the light intensity, the higher the light intensity, the higher will be the rate of photosynthesis. This is because if the light intensity is very low, there is no photosynthesis will occur due to the insufficiency of light energy, which is in form of photon, to excite the electron in the magnesium atom in the chlorophyll molecule, which is in the photosystem. So therefore, no electron transport, no ATP, and no NADPH will be formed. How about the wavelength? It depends on what pigment inside the chlorophyll, because different pigments absorb different wavelength of light. The second factor is temperature, in which the optimum temperature for photosynthesis is about 25 degrees Celsius up to 35 degrees Celsius. So above 40 degrees Celsius, the rate of photosynthesis will be decreased because the high temperature can break up the protein in enzymes such as the Rubisco and also PEP carboxylase or the other enzyme, and thus it will stop the process of photosynthesis. However, certain plants such as the Arctic algae, they work very efficient at 0 degrees Celsius. Next is is carbon dioxide concentration. The higher the carbon dioxide concentration, the higher will be the rate of photosynthesis, because we can avoid the photorespiration process to occur. So the rate of photosynthesis usually directly proportional to carbon dioxide concentration, in which carbon dioxide exceeds 1% to stimulate the closing of stomata, and it will reduce the rate of photosynthesis. And then concentrations of chlorophyll. For concentrations of chlorophyll, the higher the concentrations of the chlorophyll, it can either decrease or increase um, the concentration of chlorophyll can increase or decrease in the leaf of the plants. Okay, so the concentrations of chlorophyll will be decreased when there is a phenomena that being called as a chlorosis happen. What happened during chlorosis is that the color of the green leaf will change to a yellow color. Thus, it will reduce the photosynthetic rate because low concentrations of chlorophyll. So, when the absence of iron magnesium in the synthesis of chlorophyll and also the pigment have been destroyed by a very high light intensity, and nor it is however it is a normal aging process for leaves. Next is concentrations of oxygen. The higher concentrations of oxygen, the lower will be the rate of photosynthesis, because oxygen will compete with carbon dioxide for the active site of rubisco, thus causing the photorespiration to take place instead of Kelvin cycle. Remember, photorespiration is a wasteful reaction. And next is water availability. So the higher amount of the water, the higher will be the rate of photosynthesis, because water is important in the light reaction of the photosynthesis, which is referred to the first step of photosynthesis process. And last but not least is that the chemical compound cyanide and dichlorophenyl dimethylurea can denature enzyme that involve in photosynthesis, and also sulfur dioxide, such as the harmful gases, can also block out of the light that needed for photosynthesis. So I think that's all for today, class. Thank you.